talked about the different types of diabetes. Tonight we're going to get into a further evolution of pharmacological approaches for treating diabetes. Last week I introduced you to glucotrol. Glucotrol belongs to a class of drugs, and I'm forgetting what class that is. Anybody going to help me out? Sulfonylureas, right? And that drug that I mentioned to you is glucotrol. And glucotrol comes along and it blocks the ATP sensitive potassium ion channels. And so doing that then prevents positive charge leaving the cell. That causes the membrane potential to depolarize the threshold. When that occurs, voltage gated calcium ion channels open up and there's an increased release of insulin out into the circulatory system, which is going to help drive down blood glucose levels. Tonight we're going to talk about this class of incretins, and I'll put that word up in just a moment. And then we're going to talk about really one of the hottest things in what I'll call human biology, and that's the use of so-called GLP-like agonists, and I'll describe that for you in a moment. Really an amazing story. We see tremendous increase in glucose tolerance, or I should say glucose regulation, and a bonus to all that is a loss in weight. But these GLP agonists were made really not for weight reduction, they were made to improve the lives of diabetic patients. Okay, so that's the game plan. Once I'm done with that, then we'll kind of pick up our story that we've been going through in the lab and talking about action potentials and then how it is that cells, excitable cells, can communicate with other excitable cells. So there's this class of molecules known as incretins. And basically, this class of molecules enhances the release of insulin. And a really important discovery is that within the distal portion of the ileum, and the colon, there's a group of cells that are known as type L cells. And these are found predominantly in these regions. They're also found through other regions within the digestive system, but the highest density is in these regions here. And these type L cells are stimulated through the digestion of food in the gut to secrete a molecule known as the glucagon like peptide. And here I'm just going to put GLP. And these are amazing molecules, and they are secreted as a result of the intake of food as it moves through the digestive system. And these molecules enhance the release of insulin from the beta islet cell. They shut down the release of glucagon from the alpha islet cells. So these GLPs, they're doing exactly what I've described up there. They slow down the digestion of food in the gut. Here I'm just going to put transit. And they act on the brain, and at the level of the brain, that causes an increase in satiety. In other words, you feel full, you don't have to eat. So it reduces hunger drive. Now there's one problem with these glucagon-like peptides, and that is that they have a short half-life. So here's the brilliance of science and scientists. They understood the properties of the GLPs, and they tried to create GLP-like agonists. But the problem is that there's an enzyme in the gut that really acts on GLP-1 and degrades it very, very quickly, causing it to have a short half-life. And this enzyme in the gut that breaks down GLP is known as dipeptidyl peptase. And this is abbreviated as DPP. And there are different forms of DPP, so-called isoforms. And the one in the gut is known as DPP4. Okay, so a couple of things you've got to put together is that GLP's got this miraculous impact on the body. The problem is, in a normal state, the GLP has a very short half-life, and so the effects are fleeting. And the reason that there has such a short half-life is due to this enzyme here. And here's where the smart folks come into play. They study these proteins. This molecule, look at glucagon-like peptide. This is a peptide hormone. It's a protein. And so chemists do all types of phenomenal things. They'll shoot X-ray beams at a protein, and then they'll look at the diffraction pattern on the backside. They know the sequence of amino acids, but now they can actually figure out the three-dimensional, the spatial configuration of those amino acids. And they can also figure out what part of the glucagon-like peptide is the substrate for enzymes like this. And so what they do is they come along and they tweak these molecules. So they create a GLP-like molecule that is missing the sequence that this acts on. Everybody got that? So now the half-life of GLP is dramatically increased. Now, what's the name of this molecule that's had its structure altered such that DPP-4 won't act on it? And the name is Ozempic. There's another one, Mounjaro. So these are referred to as GLP-1 like agonists. Now this is brilliant biology, I think, is that, as I just described, they were able to identify what part of the GLP, the natural, the native GLP molecule, was being acted upon by DPP-4, and they came along and they created a agonist that looks just like GLP minus this site where DPP-4 acts. And now all of a sudden, the half-life of this Ozempic and Mounjaro is much longer in duration. And this then creates magical effects. So what's happening? Let me just finish the diagram over here. So this is the standard diagram I've laid out for you. Here we're talking about a beta islet cell. And as you already know, the beta islet cells are able to sense increases in blood glucose, because when there's increase in blood glucose, that increases the transport or the import of glucose into the cell. So the glucose levels inside the cell go up. That leads to an increase in pyruvate. There's an increase in the shuttling of pyruvate in the mitochondria. Ultimately, this leads to an increase in the concentration of ATP, and we have this ATP-sensitive potassium ion channel. And this causes this potassium ion channel to close. And when that occurs, as I've mentioned to you before, that leads to the depolarization of the cell membrane of the beta islet cell. That then causes these voltage-gated calcium ion channels to open up, and calcium enters into the cell according to its electrochemical gradient. And that calcium acts on vesicles that have insulin stored in them. So there's this common theme, somehow calcium acts as a trigger that causes vesicles to fuse with the membrane of the cell. And in this case, we've got insulin being secreted and taken up into the circulatory system and then transported throughout the body. Okay. So that's our standard scheme. So what's going on with these GLP-1-like agonists, the Ozempics, the Mounjaros of the world? Well, there's a hell of a lot of our biology that's taking place on these cell membranes. Isn't that pretty amazing? Located within the membrane of our beta islet cells are GLP receptors. So here I'm just going to put GLP-1 receptor. Here's our Ozempic. It's an agonist. It acts like glucagon, like peptide. And it comes along, and because it looks like GLP, it can bind to this receptor. So what happens? We've got this GLP receptor, and come to find out that this involves a transducer protein, a stimulatory transducer protein. You guys would call that a what? I forget what the name or the category of proteins, what we call those. A G protein, we say GS protein or G protein, that's fine. And that G protein is acting on an amplifier. And this amplifier is taking ATP and it's converting that into cyclic AMP. Not going to get into all the specifics, but here's our nucleus. Time out. Let me just take a step backwards. Insulin. What category of hormone does insulin belong to? Anybody? Don't be bashful.
Peptides?
What's that?
Peptides?
Peptides, yeah. That's the educated guess. And peptides are proteins. And what makes proteins? What codes for proteins? The genes, right? Our genes code for proteins. So here's our nucleus. We'll say that here's our insulin gene. And you guys would tell me that there's these three different regions of the genes. There's a regulatory region, there's a promoter, and then there's a coding region. So this cyclic AMP ends up creating what's called Krebs. Oop, that was good. And that stands for cyclic AMP response element. And so it binds. And it turns on the promoter. And so what's the consequence? The consequence is that now our beta islet cell is making more insulin. There are more insulin vesicles. And so if this beta islet cell now has more of these insulin vesicles, when it's stimulated, is it going to release a normal amount of insulin? Is it going to release less than normal amount of insulin or a greater amount of insulin? And the answer, I hope, is obvious to all of you, is it's going to release more insulin. Or have the capacity to release more insulin. So this dramatically impacts the ability of the body to properly regulate blood glucose levels. Just give me one moment. I want to see if I can find something here to show you. No, I don't. Oh, here we go. I'll put these out under the course content. Here we go. This is some amazing data here. This is, I think this is really exciting data. When you see stuff like this, I mean, these types of things are rarities within the field of science. So let me just explain what we're looking at here. So this is like a blood glucose tolerance test. And what they're doing is that they're coming along and they're progressively increasing the concentration of glucose in the plasma. So that's the experimental paradigm, is that they're increasing the concentration of glucose in the blood and they're seeing how the system responds based upon the amount of insulin that's being secreted. Everybody with me on that? So we've got, here's heart healthy response here in blue. Here's the response in individuals with diabetes. And you can see that there's actually very little increase in insulin secretion. Here's with Ozempic. This is after taking Ozempic for 12 weeks. And you can see that the glucose response in the diabetic individuals taking Ozempic for 12 weeks is virtually identical to a healthy individual. That's pretty darn impressive, I think. Really, really impressive. And remember, Ozempic was created to treat diabetic patients, not for weight loss. weight loss is kind of this added benefit. Now, one of the crazy things about our healthcare system and all the legal aspects of it is that for the most part, Ozempic has been shown to be pretty safe. But there's been a couple instances where individuals have developed thyroid cancer. Not statistically significant. So they're forced to sit there, and if you watch the ads on TV, there's this whole thing that runs. Ozempic may cause everything under the sun. Well, geez, there's only a gazillion people out there taking Ozempic. That's safe for the most part. It's pretty safe. Yeah.
Would it be as effective for individuals with type 1 diabetes?
No, because there we don't have the beta islet cell. So that's the problem. There's no beta islet cells, no effect of Ozempic. Okay, so that's how these GLP agonists like Ozempic work, is that they're actually acting on the beta islet cell. And as I said, the remarkable breakthrough is that these are engineered molecules that remove this sequence that the DPP4 acts on. And therefore, these beta agonists, excuse me, these GLP agonists have a long half-life. Okay, so coming back here, GLP-1 enhances the release of insulin. That's shown there. We also saw that in the data from the company itself. Decreases the release of glucagon. That's important as well. That's kind of the blood glucose side. But here's the weight side here. So if you want to try to divvy these effects up in terms of the benefit, the potential benefit, what I would say to you is that this is the diabetic side of the equation. This is the weight side of the equation. So what happens is that as the food's going through the gut, we've got these GLP-like agonists that are in the blood. And what that does is that slows down the transit of food through the gut. And so you feel full for a longer period of time. On top of that, the GLP is talking to the brain and sitting there saying, hey brain, I've got some food going through the gut. We're full down here. We don't need any more. So it's a pretty cool effect also. And this is what's led to these remarkable reductions in weight. I think I told you, we've got a family relative in Colorado. I would say that she probably was in the 250 range. She's probably in the 150 range now, so 100 pounds, yeah.
Is there like studies that people, once they come off of it, do they keep the weight off?
That's the problem, is that if you don't practice good lifestyle modifications, like regular exercise, appropriate nutrition, then we're back to thermodynamics. Energy in minus injury out, or expended, is the net balance of energy in the body, right? So if you're taking in more calories than you are expending through physical activity, then our weight's going to go up. On the other hand, if we balance those two out, then we're kind of in equilibrium. And of course, if we can somehow expend more calories than we're taking in, then we're going to have weight loss. And that's a formula that's been around forever, and it hasn't changed, really. You can sit there and talk about bariatric surgeries and all types of good things, but Ozempic is one of these things that it has a profound effect. And the problem is the one that you're addressing. Yeah.
This might be a dumb question, but just from the diabetes side of things, what is it, the benefit of Ozempic versus just actual people who are insulin dependent and take actual insulin? Because it's the
So
same
who are
effect, right?
insulin dependent?
Sure.
Yeah, so that's your question, right?
Type 2, yeah.
Yeah. So there, the issue is that you've got to have some type of a glucose monitor. You may have a glucose pump. You may not be regulating your blood glucose for the fidelity that you'd expect it to have. And when I say fidelity, I'm talking about moment by moment variation, because your beta islet cells are looking at blood glucose levels almost on an instantaneous level and varying the secretion of insulin accordingly.
And, my other question was gonna be, how, like, is there high prevalence in insulin resistance with people who are type 2 diabetic? Like, is it a common thing, or is
Yeah,
it?
it can be common. And so what creates the insulin resistance? So if insulin's being secreted,
Right.
but there's resistance on the part of the cells. What's the main cell type that takes up glucose? Skeletal muscle. If you're inactive, does your skeletal muscle need a lot of glucose? No. So I've already mentioned this. It downregulates the number of insulin receptors. By downregulating the number of insulin receptors, we downregulate the number of GLUT4s we have. So we make skeletal muscle through inactivity resistant to insulin levels. So the insulin's there, it's just that there's no receptors for it to bind to. And this comes back to, if you could take a population of diabetic patients, just through physical activity, about 35% of those individuals are going to become non-insulin dependent. That's pretty impressive, right? That's cheap. This is what I mentioned to you last week, is that just through physical activity, your body is going to correct this imbalance and properly regulate blood glucose, because the main organ that's responsible for taking up blood glucose is going to become more sensitized to it. Here, we're not talking about skeletal muscle becoming more sensitized.
Right.
We're talking about altering the way in which the beta islet cell behaves. Yeah.
Can you explain incretins again? Is type L cell a type of incretin, or is it?
No,
a
so
GLP?
incretins are a class of molecules that will enhance the release of insulin. So that's why GLP, Ozempic, those kind of fall under this category of being incretins.
Okay.
So they're molecules that enhance the release of insulin.
Okay.
Does that help?
Yes.
If you've been following the Ozempic story, when it first came out, you know it was darn expensive, really, really expensive. I want to say something on the order of $1,500 a month. $2,000? Okay, $2,000 a month. Geez, for $2,000 a month, I'm going to go get a membership in 24 Hour Fitness and LA Fitness and Orange Theory. I'm going to go hire a private trainer. So very, very expensive. The good news is that, like with everything, as more and more players get into the field, now it becomes more competitive, the prices come down. And the prices have come down substantially. And probably we'll be at a point somewhere down the road, who knows when, where something like Ozempic is going to be in the $100 range per month. That would be great. Now, the downside right now, or the big question mark, is that these molecules, the GLP agonists, are relatively new. They've only been around for 2-3 years in the clinic. And so what are the long-term consequences of being on Ozempic or Mounjaro for 5 years, 10 years? We don't know yet. So that's a big question mark. And that's why you see all this cautionary stuff comes along. Make us like and it's an association. It's not, there's nothing there that would prove that there's a cause and effect for things like that. I don't want to trivialize that, but because you always want to be conservative. But this is a pretty remarkable group of molecules. The other thing that I want to impress upon you is that this is taking away the obscurity in terms of how we regulate hunger. We regulate hunger around things like GLP-1 telling the brain, hey, we're full, we don't need any more food in the gut. And the gut is also sitting there telling the brain, hey, we're full, no more food, please. All right, so that's that. So we've got our sulfonylureas, glucotrol, an example. We've got our GLP-1 agonists like Ozempic and Mounjaro. What else? What's another common GLP-1 out there? Anybody? Wegovy, yes. I was going to write Wegovy, but that's another one out there. And there's got to be some others. Anyways, I'll send you that graph that I had up there. I'll send you the URL for that. You can look at that. Now, there's another group of molecules. Biguanidines, really a very, very important class of drugs used to treat diabetes. I want to make sure I'm spelling that right, so just bear with me here for a second. Yep. And here's the big gun in terms of drugs that falls under this is metformin. And this goes by a generic name known as glucophage. Yeah, I want to note for you that this is such an important class of drugs, especially metformin, that this is in the top 100 drugs listed by the World Health Organization. So the World Health Organization's got this list of the 100 most important drugs that any hospital should have. So there's a whole list for cardiac drugs. With regards to diabetes, this is a big one right here. So here I'm going to put WHO. That stands for the World Health Organization, top 100 drugs. And metformin leads to a decrease in the release of glucagon, causes an increase in the secretion of insulin, and decreases the conversion of amino acids into glucose. Yes. The other thing I need to mention here is that within the liver, so in the liver, remember the liver is where we're storing glucose in the form of glycogen. So metformin decreases glycogenolysis. So here's that word, glycogen, and then lysis, the breakdown of glycogen and glucagon causes the hepatocytes to increase glycogenolysis. So here, metformin is impairing that. Okay, any issues that we need to clear up here? With diabetic patients, there's this terrible condition that can occur, and this is known as diabetic ketoacidosis. Well, hopefully when you look at this, or as you're writing it down, you see that word, acidosis. So this is a condition where the pH of the blood is altered and becomes acidotic. And this acidosis occurs due to the breakdown of lipids into so-called ketone bodies. And these ketone bodies then give rise to ketoacidosis. So in this situation, there is a decrease in glucose metabolism, and so that's going to increase the dependence upon which fuel? What are the two primary fuels that cells use? Two primary fuels that cells use. Two primary fuels that cells, the fuel to make ATP. Glucose and lipids, triglycerides. Well, if we have a decrease in glucose, then we're not going to be able to use glucose, so that means that we're more reliant upon these fatty acids. Test question time. Ooh, let's see here. Camilla, where's Camilla? Camilla? Okay, so this is stuff we've talked about before. So in these individuals, there's a decrease in glucose. We have this increased use of triglycerides as our fuel source to make ATP. What's going to happen to the respiratory quotient in these individuals? You can pass? Did you want to pass to Eric?
Sure.
You can pass. You can pass to Kaylee if you want, or to Bennett.
That's the case.
Can you restate the question, please?
Yes, what's going to happen to the respiratory quotient in these individuals? Go for it.
I think it's going to drop to 0.7.
That's correct. That's exactly right. So we've got this patient who's having this crisis, and they show up. If you had the wherewithal, you wouldn't do this clinically, but if you had the wherewithal, you could come along, sit there and say, okay, I'm just going to collect their gases, and oh, by the way, I'm measuring the CO2 output and the oxygen consumption, and I can see that the respiratory quotient's at 0.7 or 0.71 or 0.72. That means virtually all the ATP is being produced that way. Well, being the good biochemist that you are, you know that this triglyceride is broken down in a molecule of glycerol, and that molecule of glycerol can be fed into glycolysis, right? You guys are all going to go back and review your notes on this. And the fatty acids are broken down through a process known as what? How do they enter into the Krebs cycle? What's the process? Beta oxidation, that's right. And so with this increase in fatty acid utilization, we have an increase in what are known as ketone bodies. Ketone bodies are the result of metabolizing fats. Look at the term up here, diabetic ketoacidosis. Keto, that's in relationship to the metabolism of fats that end up producing ketone bodies, which make the blood acidic. So that causes a decrease in pH. Well, let me first come along, say that it causes an increase in the concentration of hydrogen ions in the plasma. Now for this next part, we have to turn to our resident biochemist, Elizabeth. She'd probably go, he's not going to call me tonight. Surprise, surprise. Well, you're sitting next to a biochemist, so shame on you. Sorry about that. Let me take my watch off here. Elizabeth, what is a pH scale?
I mean, it's kind of a measure of the concentration of hydrogen atoms and hydroxide ions, sorry, hydrogen ions and hydrogen ions.
Good so far, I like it. Except, we need to add one part to it, and that is the pH scale is a inverse log scale of the concentration of hydrogen ions. So as the concentration of hydrogen ions goes up, since it's an inverse scale, that means that the pH is going down. By the way, this is a life-threatening phenomenon here. This isn't something you just sit there and go, oh yeah, I got ketoacidosis, I think I'll walk around for the next 2 days or 3 days. You're going to feel so darn bad that you're going to sit there and say, hey, get me to the emergency room. This is an emergency room condition here. So this leads to a decrease in pH. And I'm just going to put arterial pH. Here's something I want to plant the seed regarding the importance of pH. Is that the arterial pH determines the pH of the rest of the body. We're going to make it just a simple rule. So the rule is the arterial pH affects the pH everywhere in the body. Because those hydrogen ions are escaping out of the circulatory system and going into the interstitial fluid. They're right there, right around all the cells in our body. That's weird. Austin, where's Austin? No Austin. Yes, let's see here. Is there here? Let's see. Camilla. Miss Camilla. I see you back there. Why do we care about the concentration of hydrogen ions? Do you know anything about protein chemistry? If you don't, no harm, no foul. You can pass to Miss E, and if she doesn't know, she can pass to Mr. K.
Well, protein, like protein in your urine can be a sign of, we wanna check your blood pressure after that.
Okay?
Cause it can be a sign of hypertension.
All right.
So if there is protein in your urine, that's not a good thing. So that's what I know about protein.
Okay, all right. So my question is this, why the heck do we care about the concentration of hydrogen ions? So what? Who cares? I don't care. We shouldn't care. Nobody in here cares. You care?
I think so.
No, I'm talking to Miss E back there.
I mean, yeah.
What's that?
Yeah, I mean, when we were talking, just like in a general sense, the proton pump to generate
Okay.
ATP.
All right, okay. Mr. K, what would you say, being a biochemist? Why do we care about the concentration of hydrogen ions?
Because.
Does this have anything to do with the first night in our class, by any chance?
You have to refresh me when it comes to the first name.
Oh, no, I'm not gonna
Well, I can
refresh your
answer the
mind on that.
question.
No.
What
No.
structure
As a matter of fact, let me make a note right here. Matt needs a refresher. Got it, okay.
When it comes to structure determines function,
There we go. Now we're on to the right track.
the R groups of the amino acids, they have a certain structure to them. But where there's too many hydrogen ions, they will interact with that R group, changing the structure to have an extra hydrogen, then changing the function. And then
Of these
of
proteins,
these
right?
proteins, leading
Yes,
to unfolding, etc.
that's right. Structure does determine function. And as it turns out, concentration of hydrogen ions and what else? What other common thing affects the structure of proteins? Starts with a T, ends with an E.
The temperature.
Temperature, that's right. And some people use the term denaturing, the unfolding, if you will, of proteins. So this is cool. This ties right back into our first night in this class where I presented to you principle number 1, structure determines function. And as it turns out, the concentration of hydrogen ions determines the structure of proteins. Proteins are the machinery of our cells. And if we alter their structure, we're going to alter our physiology. In this case, not in a good way. So this is kind of this cascade of ketoacidosis. By the way, this oftentimes you'll hear this described using the following abbreviation, DKA. That is an emergent condition. Okay, I think that's where we're going to stop here. Let me just make sure. There's one other thing I need to mention. The triad of DKA. Bless you.
Thank you.
Let's see.
Glycemia. I'll put hyper. Hyperlipidemia. And hyper that's the so-called triad of DKA. How many of you in here know of somebody, friend, family member, who's on one of the GLPs? Anybody? I already told you I've got 1, 2, nobody else, huh? 3, okay. All right, so now we're transitioning away from the endocrine system, and we're gonna come back to neurophysiology. And so last week we left off talking about the propagation of action potentials down along the length of the axon. And here I'm just gonna show what I want you to interpret as a dendrite from, yes, go ahead.
Sorry, for the glycemia, is that
Hyper. So we're gonna say that this is a dendrite. And the way I'm gonna describe this is we're gonna say that the flow of information is flowing down along this cell here into this cell here. Tonight, if I asked you, what types of key membrane proteins are found along the length of the axon? Your answer, you sit there and say, leaky potassium ion channels, sodium potassium ATPase pumps, voltage gated sodium ion channels, voltage gated potassium ion channels. I'm just going to show the voltage gated sodium and potassium ion channels. And I'm just noting those through red for potassium, and blue for voltage gated sodium ion channels. There's a diagram that I made for you upstairs that showed the distribution of various membrane proteins all along the length of a neuron. Make sure you know that. Those are easy points. You should never lose points on questions related to that. And these are distributed all down along the length of the axon and into the membrane of the axon terminal. Are there voltage-gated calcium ion channels along the length of the axon? The answer to that is no, there are not. Where are voltage-gated calcium ion channels located? The answer is the membrane of the axon terminal. So I'm just going to use green to represent our voltage-gated calcium ion channels. And located within the axon terminal, we've got these vesicles. And the molecules that are going to be used to talk to this other cell, we're going to refer to those as neurotransmitters. So we're going to call this vesicle a neurotransmitter. Vesicle. And I'll just put little red dots in here just to represent the presence of neurotransmitter. Okay, so if everybody would just look over the board for a moment. I need everybody, all eyes up here. So out over here to the left, I haven't drawn it, but we've got the dendrites, we've got the soma of this cell. And the dendrites over here allow positive charge into the cell. That positive charge gets the initial region of the axon to threshold. And now you would tell me that the voltage gated sodium ion channels in a given region open up in a all or none phenomenon. And that causes the adjoining membrane to reach threshold, where all the voltage gated sodium ion channels open up in all or none, which causes the adjoining membrane to open up, all the voltage gated sodium ion channels to open up, which causes the adjoining region to get threshold and all the voltage gated sodium ion channels open up. And this process of renewing the action potential as it spreads down along the length of that axon is known as what? Say louder.
Propagation.
The propagation of the action potential, the renewal of the action potential. So here I'm just going to put an arrow. Here I'm just going to put propagation. Of the action potential. That then causes the membrane of the axon terminal to reach threshold, which causes the voltage gated sodium ion channels there to open up in a all or none phenomenon. And the depolarization of the membrane of the axon terminal causes these voltage gated calcium ion channels to open up. And now calcium comes flowing into the cell, and all of you are going to tell me calcium's going to come in according to its electrochemical gradient. Within the membrane of the axon terminal, this is an amazing part of the story, there are little docking proteins. Sit there and think about your boat. How do you tie up at shore? You go to a dock. How do these vesicles unload their chemical messengers? They got to tie up at a dock. How they do that? Through protein-protein interaction. So here I'm just going to show this protein, and we're going to say that this little indentation is the docking site. Is there only one of these? Of course not. There's a lot of them. On the membrane of our neurotransmitter vesicle, there are proteins that are complementary to that docking site. I'm not going to do all of them here. But what happens is that when calcium comes in, it acts on some protein kinases. And that then causes this vesicle to fuse. Here I'm just going to write fusion. To fuse with the membrane of the axon terminal. And when that happens, the vesicle, the membrane of the vesicle, since it's a phospholipid bilayer, it joins into or becomes part of the phospholipid bilayer of the membrane of the axon terminal. And that allows it to release its chemical messenger out into this gap, this space. What's the name of that space between these two neurons?
Synaptic gap.
That's the synaptic
Gap.
synaptic cleft.
Oh.
what's the name that we give or use to describe the connection between these two cells? The answer is right up on the board. What's the name that we use to describe the connection between these two cells? Starts with an S, ends with an E. Say it louder. Synapse, that's right. So this is, remember, earlier I mentioned to you 6 different ways in which cells communicate with one another. Autocrine, paracrine, neuroendocrine, endocrine, and those were the 4 that I drew. And then there are gap junctions and there's synaptic forms of cell-to-cell communication. Here's our synapse. All right, now we're going to refer to this cell to the left as the presynaptic cell. Well, if that's called a presynaptic cell, then all you're going to tell me that the cell to the right here is the postsynaptic cell. Let's say that within the membrane of the dendrite of this postsynaptic cell, that there are these receptors, and we're going to refer to this one, since I've shown it in blue, we're going to call this a receptor gated, so RG, receptor gated. That's opposed to VG, voltage gated. So when we think about ion channels, they can be gated either through voltage changes or through binding ligands. And here we have a receptor gated ion channel, except I'm going to call this a receptor gated sodium ion channel. Well, when this neurotransmitter binds to the ligand binding site, that's going to cause the ion channel to open up. And now you would tell me that sodium ions will flow into this cell according to, I say this ad nauseum, the electrochemical gradient for sodium. And we're just going to say, to keep things consistent, we're going to say that in this postsynaptic cell, the resting membrane potential is minus 70 millivolts. Let's see here, I'm thinking of a name, yes. Have I called on Camilla yet today? No? We're in the land of Logic 101. You should be able to get this. So we've got these receptor-gated sodium ion channels open up. Now, I need all of you to look at that diagram that you drew. This is from lab, where we have the distribution of key membrane proteins along the length of a neuron. Do you guys have receptor-gated ion channels at the level of the dendrites? So please appreciate this. Our receptor-gated ion channels are in the dendrites. Our voltage-gated ion channels are along the length of the axon and the axon terminal. So Miss Camilla, back to you now. You thought I was going to forget about you, huh? When that sodium comes flowing into the cell, what's going to happen to the membrane potential inside that cell? Is it going to stay at minus 70 millivolts? Is it going to hyperpolarize, in other words, become more negative? Or is it going to depolarize? What would you say? It's going to depolarize. And if we get the axon hillock, which is where we have the first presence of voltage-gated sodium ion channels, if we get the membrane and the axon hillock to threshold, then you're going to tell me that the voltage-gated sodium ion channels all along the length of the axon are going to open up in what type of a manner? Okay. Huh? No. All or none. That's it. All or none. By the way, this all or none phenomenon during the depolarization phase, is that a positive feedback or is that a negative feedback? It is a positive feedback because once I get one voltage gated sodium ion channel to open up, they're all opening, right? Because the rule is all or none, right? All or none. All I got to do is get one of them to threshold, they're all going to open up. That's a positive feedback loop. But I mentioned something to you, and that is positive feedback loops are very short-lived because that's not consistent with homeostasis. All right, Camilla, this event that we produce over here, will it be inhibitory? Will it be nothing, or will it be excitatory? That's right, it will become excitatory. And so what we're going to say is that the binding of this neurotransmitter to this receptor-gated sodium ion channel is going to produce a response in the postsynaptic cell known as an excitatory postsynaptic potential, an EPSP. So here I'm going to just write EPSP. EPSP. That's a lot of words, so using EPSP is a much friendlier way to talk about these things. Here's a receptor-gated potassium ion channel. All right. Let's see. Kaylee, you're up to bed on this one.
Mm-hmm.
So this neurotransmitter binds to this receptor-gated potassium ion channel, and you're going to tell me that potassium ions are going to move into the cell, causing the membrane potential to go up and creating an EPSP. What do you think? How do I do? Sounds right, doesn't it? How many of you guys ever used to watch? Oh, I know you guys don't watch TV anymore. There was a show on back, it was like 50 years ago, called The X-Files. I know none of you watched that. But a lot of The X-Files was about aliens. Now, all of you know that outer space aliens landed in Roswell, New Mexico in 1958. That's a fact. What are you laughing about? And I have to forewarn you, aliens can show up on my tests. So, the proper way to analyze this problem is that, I forget, what's the concentration of potassium inside the cell, young lady?
Oh, wait, concentration of potassium, isn't it 4 millimoles?
Where?
No, wait, oh, it's 140.
140 outside the cell, right?
Yep. No, isn't it inside, right?
Okay, whatever you want to tell me, I'm just
Yeah.
gonna write what you tell
I'm
me.
gonna say, right? Yeah, yep, inside. And then it's gonna be 4 millimolar outside of the cell. So then it would be flowing out.
So just so I make this right, so potassium, the concentration of potassium inside the cell is 140 millimolar, and you're saying the concentration of potassium is what?
4
I don't know, is it 4 or is it 44?
4 millimolar.
Is it, well, you guys said it again, you said
4.
44 again.
4.
Just 1 4.
Yeah, just 1 4. And so then that means potassium would be flowing outside into the synaptic cleft.
So the arrow should go that way.
Yes.
But wait a minute. What's the membrane potential inside the cell?
Negative 70 millimolar.
Hmm?
Isn't it negative 70?
Negative 70 millimolar, is that what I heard you say?
Millivolts.
That's correct. It is minus 70 millivolts. So there's a chemical gradient that does favor the efflux of potassium out of the cell. This is, I hope, reinforcing for you these types of questions. The alien, which I've studied at Roswell intensely, can show up on test number 2. So you just have to go through the steps. The electrical gradient favors the movement of potassium in which direction?
Inside.
Out of the cell, right?
Yep. Yep.
Because Coulomb's law says that like charges attract and opposite charges repel. Okay. Coulomb's law does not say that. Coulomb's law says that opposite charges attract and like charges repel. The inside of the cell is minus 70 millivolts. The charge on potassium is positive. Opposite charges attract. So the electrical gradient doesn't favor the efflux of potassium. It favors the influx of potassium. How do I know which of these two forces is greater? And your answer is, well, Vince, I know that Nernst's equation tells me what the equilibrium potential is for any given ion. And the equilibrium potential is one the electrical gradient is exactly equal to but opposite the chemical gradient. The equilibrium potential for potassium is what? Minus 92, minus 90 millivolts. We're not there. So the electrical gradient is not strong enough to cancel out the chemical gradient. So will there be an efflux of potassium? Yes, it will. And that efflux is going to cause the cell to depolarize, to stay at minus 70 millivolts, or to hyperpolarize. It will hyperpolarize. Are we going to call that an excitatory postsynaptic potential, or would you call that an inhibitory postsynaptic membrane potential? That's what I would call it, an IPSP. Okay, unfortunately, my time has run over. If you have questions about this, come on up. I'm happy to try to answer those. Otherwise, we'll be picking the story up on Wednesday night. Study hard, Make sure you learn that Nernst equation.